BPS Bioscience offers tools and services to support your drug discovery research. In this video, we will go over how to thaw frozen cells.
Pre-warm the thaw medium for 10 minutes prior to thawing your cells. Disinfect the bottle of medium before moving it to a laminar flow hood. Pipette 10 milliliters of medium into a conical tube. After removing your vial of frozen cells from liquid nitrogen storage, make sure to keep it on dry ice until you are ready to thaw. Swirl the vial of frozen cells for approximately 60 seconds in a 37 degrees Celsius water bath. As soon as the cells thaw, disinfect and move them quickly to the flow laminar hood. Transfer the contents of the vial into the tube containing 10 milliliters of medium. Immediately spin down the cells at 300 G for 5 minutes. Remove medium by aspiration, being very careful to not disturb the cell pellet at the bottom of the tube. Resuspend the pellet in 5 milliliters of fresh, pre-warmed thaw medium. Note that the thaw medium does not contain selection antibiotics. Transfer the resuspended cells to a T25 flask and move it into an incubator at 37 degrees Celsius in 5% carbon dioxide. At 24 hours of culture, check for cell health and viability.
For more products and services to support your drug research and discovery efforts, visit bpsbioscience.com.